डोंट ओपन द बुक, ओके। ये बुक है, जब क्लास हो जाए ना उसके बाद जाके देखना क्या छूटा, क्या पढ़ाया। So day before yesterday we have started with the renal system, okay. We have started, we have done something related to the renal system, and we have completed, tried to complete the basic structural unit of kidney, okay. We have started with the functions of kidney, and the functions were categorized into excretory functions and non-excretory functions. You get a short note on non-excretory functions of kidney. And what were the 4 important non-excretory functions? Homeostasis can be further divided into the 4 main components of homeostatic mechanism. One was, anyone? कोई भी एक फंक्शन, होम्योस्टेसिस बहुत सारी चीजों को इन्वॉल्व करता है, कोई भी एक फंक्शन। टेम्परेचर को रीनल में कहाँ रोल आ रहा है? pH regulation, okay. One of the functions was pH regulation, another function was osmolarity, okay. Third function बच्चे रीनल में टेम्परेचर रेगुलेशन नहीं आ रहा ना, अपन होम्योस्टैटिक मैकेनिज्म की बात कर रहे थे, उसमें 4 जो फंक्शंस हैं होम्योस्टेसिस से रिलेटेड वो क्या थे? उसमें से इसने ऑस्मोलरिटी बता दिया, उसने पीएच रेगुलेशन बता दिया, क्या बच रहा है? आयनिक बैलेंस। बैठो। बॉडी फ्लूइड वॉल्यूम, ओके। So they are further, और उसके बाद में synthesis, secretion and metabolism of various hormones, okay, resulting into the regulation of blood pressure or arterial blood pressure. So regarding the functions of blood pressure regulation, we need to have the renal circulation idea, okay. So in case of renal circulation, the total cardiac output comes out to be 5 liters. So approximately 1 liter or 20% of the total cardiac output is coming to the renal circulation. Through the descending aorta branch, okay, it will further divide into the renal artery. On both the kidneys it will go, and further it will be divided into different layers, okay. Now final capillaries which we are concerned, this is the only place in the body which we are getting a double capillary network. Only the system in which the capillaries are having the highest hydrostatic pressure. इधर इधर खड़े हो जाओ। You are using your phone? तुम्हारे पास वाला ब्राउन स्वेटर, तुमसे पीछे वाला। मैं तुम्हें 10 बार बता चुकी हूँ, यहाँ से एक एक जने की चीज हम देख सकते हैं और ऑब्जर्व कर सकते हैं, ठीक है। Keep it in a silent mode, okay. Your father knows that you are present in college, okay. So the main arteries which are responsible for this is the renal artery that will enter into the kidneys by the bifurcate, interlobar, arcuate, interlobular, resulting into the efferent arterioles. Efferent arterioles are further divided into the tuft of capillaries which we call as glomerular capillaries, and this glomerular capillaries will finally result into efferent arterioles. Efferent arteriole finally results into the peritubular capillaries or vasa recta. So they have a double set of capillary networks, and the pressure present in the capillaries compared to elsewhere in the body is highest because the function they have to perform is the function of ultrafiltration. Okay. Now this renal circulation, is responsible for regulation of blood pressure. तो renal circulation को दो तरह से कंट्रोल किया जाता है. भाई physiologically there might be variation in the renal circulation. There might be physiological variation in the blood pressure. We are continuously seeing that the posture also results into change in blood pressure, exercise, day-to-day activity. So all these things, they change the blood pressure. But kidneys have to regulate this system, plus, okay, the circulation has to be maintained. So all these work simultaneously, and for this renal circulation mechanism, two probable theories are given which are responsible for maintaining this 20% of the total cardiac output or 1 liter of the blood that is coming from the cardiac output. Now, according to the position of the nephrons, okay, they are further classified into two types of nephrons. They are the cortical and the juxtamedullary nephron. मैंने कहा था कि मैं लास्ट क्लास में आप पढ़ के आएंगे. ठीक है. चल. Then we are calling it as cortical nephron. Okay. The number of cortical nephron in percentage is, it is 80 to 85%, and the juxtamedullary nephron is 15 to 20%. What is the most important function performed by the cortical nephron? Filtration, reabsorption, and no. It is only formation of urine. Okay, the most important nephrons which are involved in the formation of urine are the cortical nephron. Now the steps of formation of urine is further classified into filtration, reabsorption, and secretion. It is not excretion. Excretion and secretion vary. So it is filtration, reabsorption, and secretion. These are the three main steps which will be responsible for the mechanism of urine formation. If the question comes boldly, you have to write down the three main steps involved in urine formation are ultrafiltration, reabsorption, secretion. Yeah, definitely you are going to get 2 marks. Okay, so cortical nephrons, they are responsible for the formation of urine, and juxtamedullary nephrons, they are responsible for the concentration mechanism or maintenance of osmolarity. So the homeostatic mechanism का एक part जो अपन ने पढ़ा था वो था osmolarity maintenance, and that osmolarity maintenance is done by the help of juxtamedullary nephron, because they are having some variation in the structure of the nephron compared to the cortical nephron. Now this variation in functions depend on the structure of the nephrons which are present in cortical region and in the juxtamedullary region. What are the main parts? When we are talking of the cortical nephrons, the glomerulus or the renal corpuscle or the Malpighian body, they are present in the cortex region, so appear granular in, when you see the structure, they appear granular. Okay. In case of juxtamedullary, they are at the junction of the cortex and medulla. Okay. When we are talking of the cortical nephrons, they have another set of capillary networks that is known as peritubular capillaries. Whereas when we are talking of the juxtamedullary nephrons, they have another specialized type of capillary networks. They are known as vasa recta, which is mainly responsible for exchange mechanism that will result into the either the concentration of the urine or dilution of the urine depending on the osmolarity or the water intake done by the body. So that is done by the juxtamedullary nephron. When we are talking of the parts of the nephron, the Malpighian corpuscle structure is same in both the nephrons. When you are talking of the PCP segment, it is same in both the segments. Okay, but when you are talking of the loop of Henle, the descending segment is same, but the length of loop of Henle is more in juxtamedullary nephron compared to the cortical nephron. Another difference is when we are talking of the ascending segment of loop of Henle, there are thin and thick segments. Okay, and then it comes into the distal convoluted tubule, collecting tubules, and collecting duct. Clear? Now, if you study in detail, अपने पास में जब अपन molecular level पे पहुँचते हैं ना, तब अपन थोड़ा सा pharmacology और medicine भी देख सकते हैं. Right now, you will be more interested in doing what is required for just getting marks or what कि अपन first MBBS निकाल लें, है ना? वो क्राइटेरिया लेके चलोगे तो थोड़ा सा आपको ये बोरिंग लगेगी, बट थोड़ा सा आप उसमें हेल्प अपने आप को थोड़ा सा एक्टिवली इन्वॉल्व रखना तो थोड़ा सा इंटरेस्टिंग हो जाएगा. एक्सक्रीटरी सिस्टम के अंदर या रीनल सिस्टम के अंदर द बॉडी इज फिल्ट्रेटिंग राउंड अबाउट हाउ मच ब्लड इट इज फिल्ट्रेटिंग 1000 to 1200 ml per minute. So per day it is filtrating 180 liters per day. But out of 180 liters, only the normal excretion of the urine is 1 to 1.5 liters. So what is the rate of urine formation? Rate of urine formation. It is 1 ml per minute. You calculate it and you will get the value 1 to 1.5 liters. So the rate of urine formation is 1 ml per minute. 125 is the rate at which all the glomerulus present in both the kidneys are able to filter blood. So when you are writing the answer mechanism of urine formation, okay, some bright students write that the urine is getting filtered. It is not urine, it is blood which is getting filtered. ठीक है, तो फर्स्ट लाइन ही आपकी गलत हो जाती है तो सारा आंसर ही गलत होगा. ठीक है, ये सालों से होती हुई गलती है जो मैं बता रही हूँ कि ये रिपीटेडली होती है चीजें. ठीक है, तो फर्स्ट स्टेप इज अल्ट्रा फिल्ट्रेशन. पर फिल्ट्रेशन किसका हो रहा है? ब्लड का. एंड per day how much blood is getting filtered? 180 liters. When we are talking in minutes, it is 1200 ml per minute. Clear? तो ये 2-3 वैल्यूज है ना, इनको बिल्कुल घोट के जैसे पी लेते हैं ना, क्योंकि वाइवा के एक क्वेश्चन में आपकी जिंदगी आर पार हो जाती है. ठीक है, तो ये कुछ वैल्यूज हैं जिनको कि हमें लिख के याद रखना है. Clear? And continuation with the structures. In case of females, the excretory system, the part from the ureter to the bladder to the urethra is small compared to the males. Okay, so females are more prone for urinary tract infections. Okay, when we are talking of the nerve supply to the kidneys, okay, kidneys are getting only the sympathetic nerve supply. They are not getting the parasympathetic nerve supply. The parasympathetic nerve supply is coming to the bladder. It is not going to the kidneys. So sympathetic innervation going to the renal system, but parasympathetic and sympathetic both coming to the bladder, and they are both responsible along with the somatic nerve for the micturition reflex. What is micturition reflex? It is the voiding up of urine when the bladder gets filled upper to its capacity. First micturition reflex sets when the bladder is filled with 300 ml of. This is cystometrogram. Last row. If you are not interested, you can move out. Okay. Another important questions which you got is, as short note is, write a note on cystometrogram, and more than 50% of the students, they relate the cystogram with the cyst present in the ovaries. Okay, it is not cyst present in the ovaries, it is the pressure volume change taking place in the urinary bladder, and the study of this pressure volume change in the bladder is known as cystometrogram. ठीक है, कुछ भी लिखना है, इसका मतलब कुछ भी नहीं लिखना. Cyst, it is not always ovarian cyst. Okay, what you commonly hear, it is the cystometrogram or the study of the pressure volume changes taking place in the urinary bladder. Okay, that is cystometrogram. So when we are going to do the micturition reflex, there we will see it in detail. Now, basic mechanism of urine formation. What are the basic mechanisms which are taking place? The afferent and the efferent arterioles. Okay, when you are making a diagram, please make it a bold presentation that there is variation in the diameter of the afferent and the efferent arterioles. Okay, so the concept, आपका concept भी clear हो जाता है और हमें भी easy हो जाता है. We can say that the person is having a clarity regarding the functioning of the nephron. Okay, so the diameter varies of afferent and efferent arterioles. Diameter of the afferent arterioles is more compared to the efferent arterioles. What is the total length of nephron? Second last row. आ जाओ आगे. Black. आ जाओ आगे. आ जाओ. What is the total length of nephron? It is 45 to 65 millimeters. Length of total of a single nephron is 45 to 65 millimeters. For our convenience, we are dividing it and seeing it in a particular fashion कि भाई इस तरह का structure है, but they are very much closely packed. Okay, so along with the vessels and they are closely packed. तो देखो इस तरह का नहीं दिखता, but for convenience we are dividing and we are showing you the structure of the nephron. Okay, now what are the basic functions which are involved in mechanism of urine formation is, that is the filtration, reabsorption, secretion and finally excretion. So whatever the blood is coming through the afferent arterioles resulting into the glomerular capillaries is getting filtered. Okay, it is getting maximum reabsorbed. Some of the substances which are unwanted in the blood, they are getting secreted and finally they are getting excreted through the urinary system or the excretory system. Okay, now this filtration, okay, why we are calling it ultrafiltration? बताओ. दूसरी बात कैसे, कहाँ पे हो रहा है ब्लड का फिल्ट्रेशन पहले? हाँ हाँ, नहीं, जो ग्लोमेरुलर कैपिलरीज है, ग्लोमेरुलर क्या है? ग्लोमेरुलर एक सेट ऑफ टफ्ट ऑफ कैपिलरीज है ना, और बोमेन्स कैप्सूल क्या है? एक ब्लाइंड बिगनिंग है नेफ्रॉन की. ठीक है, तो इनका कॉम्बिनेशन से एक ऐसा स्ट्रक्चर बनता है दैट इज नोन एज फिल्ट्रेटिंग मेम्ब्रेन. तो फिल्टर तो ब्लड एक ही बार हो रहा है, बट द प्रेशर विच इज रिस्पॉन्सिबल फॉर द फिल्ट्रेशन इज tremendously high compared to the pressure which is present in the capillaries elsewhere of the body. जहाँ पे भी कैपिलरीज है बॉडी के अंदर, उसमें इतना प्रेशर नहीं है जितना कि ग्लोमेरुलर कैपिलरीज के अंदर प्रेशर है. सेकेंड पॉइंट है कि इसके अंदर नैनो मीटर के डायमेंशंस के पार्टिकल ही फिल्टर हो पाते हैं. तो इस कारण क्या होता है, फिल्ट्रेटिंग मेम्ब्रेन के 3 स्ट्रक्चर हैं, उसमें से फिल्टर होके निकल रहा है. सो वी आर कॉलिंग इट एज अल्ट्रा फिल्ट्रेशन. फिल्ट्रेशन तो एक ही बार होगा, ठीक है, पर ग्लोमेरुलर कैपिलरीज, बोमेन्स कैप्सूल और उसके कॉम्बिनेशन से बीच में जो मेम्ब्रेन है एक फिल्ट्रेटिंग मेम्ब्रेन बनाती है. दैट विल रिजल्ट और गिव यू द वैल्यू ऑफ फिल्ट्रेशन कोएफिशिएंट. ओके, फिल्ट्रेट क्या हो रहा है? टोटल कितना ब्लड आ रहा था अपने पास फिल्ट्रेशन के लिए? 1200 ml. ओके, composition of blood is plasma. How much quantity is plasma? So round about 650 ml of the blood plasma is getting filtered. तो अब अपन उसको रीनल ब्लड फ्लो बोलेंगे, पर फिल्टर क्या हो रहा है? रीनल प्लाज्मा. So renal blood flow is 1200 ml, but what we are filtering is renal plasma, and the amount or the quantity of renal plasma comes out to be 650 ml. Reabsorption 180 लीटर्स फिल्टर हो रहा है, पर एक्सक्रीट कितना हो रहा है? केवल 1 to 1.5. So 99.9% they are getting reabsorbed. Okay, they are getting reabsorbed, and some of the substances like drugs, toxins, metals, they have to be, and the hormones, or they are to be removed from the body, so they are getting secreted and finally getting excreted. तो चार टर्म्स है, पर चार टर्म्स की क्लियरिटी होनी चाहिए. फिल्ट्रेशन, रीएब्जॉर्प्शन से आप क्या समझ रहे हैं? फिल्ट्रेट में जो आ रहा है उसका पेरिट्यूबुलर कैपिलरीज या वाजा रेक्टा वापस रीएब्जॉर्ब करें. अपन इसको एब्जॉर्प्शन भी बोल सकते थे, पर अपन इसको रीएब्जॉर्प्शन, री मीन्स अगेन. Previously they were present in the blood, now they getting entered again into the blood component via maybe peritubular capillaries or through the vasa recta. So the mechanism is known as reabsorption. And some of the substances, if the pH increases or decreases, so the hydrogen ion or the bicarbonate ion may enter, that is they are secreted and they enter into the filtrate. And finally they are eliminated or excreted. So these are the steps. Renal handling of different substances. What happened to the different substances which are getting filtered? Okay, some substances they are only getting filtered. Finally, इनका ना reabsorption हो रहा है, ठीक है, और ना इनका secretion हो रहा है, कुछ भी नहीं हो रहा है, तो total amount जो filter हो रहा है वो urine में आ रहा है. Some substances they are getting filtered, but they are getting partly reabsorbed. Some substances they are getting filtration, complete reabsorption, not at all entering into the urine. Substances which are getting filtration is done, but they are added. इसका हेडेक हो रहा है? आप बाहर जाओ बाहर जाओ प्लीज. इतना लोड मत लो. So what can be the example of the substances which are getting filtered and excreted finally? अरे तो तबीयत खराब होती है तो मत आया करो ना बच्चे. किसने बोला है? देखो हेल्थ इज वेल्थ. क्यों सिरदर्द ऐसे तुम्हारे कहने से नहीं होगा? नहीं, इस तरह से टॉर्चर मत दो. इतनी टॉर्चरस क्लास है तो मत आया करो बच्चे. ऐसा कोई आपको लगता है कि ये बहुत टॉर्चरस है तो मत आओ. केवल पास अपने को एनाटॉमी में तो होना नहीं होता है ना. इधर बैठ जाओ, इधर नहीं होगा. So some of the substances which are getting filtered and finally getting excreted, neither they are getting reabsorbed, neither they are getting secreted. Okay, example is creatinine. If the substance which has to be, this creatinine estimation is a gold standard test. Another test for this gold standard test is para-amino uric acid that has to be infused. So that is a separate story, but naturally what substance is getting filtered and excreted, neither getting secreted, neither getting absorbed, is the example is creatinine. Another example is the substances they are getting filtered, but they are getting reabsorbed. So some most important example is water. सबसे बढ़िया एग्जाम्पल इसका क्या है? वाटर. ओके, सबसे ज्यादा क्वांटिटी में क्या रीअब्सॉर्ब हो रहा है? वाटर. वाटर के बाद में ग्लूकोज, ठीक है, सोडियम, क्लोराइड, आयंस, प्रोटीन्स, they are getting reabsorbed. But maximum amount या क्वांटिटी किसकी है? वाटर. ठीक है, फिल्ट्रेशन हो रहा है, कंप्लीट रीएब्सॉर्प्शन हो रहा है. कौन से होंगे ये? ग्लूकोज, ठीक है, ग्लूकोज, अमाइनो एसिड. ओके, दे आर द सब्सटेंसेस normally physiologically they have to be reabsorbed. If they are not getting reabsorbed totally, there it results into pathological condition. And fourth is secretion of hydroxyline, secretion of some drugs, secretion of toxins. Okay, some of the waste products which are made to be excreted with the help of combination of the water soluble complexes, and they are getting added to the filtrate to be removed or voided by the excretory. when we are seeing water, so 180 में से 179 liters is getting reabsorbed. Okay, sodium out of this millimoles, only 150 is millimoles is getting excreted. Glucose out of 180, 180 is getting reabsorbed. Creatinine 1.8 is filtered and that is totally excreted. Okay, next topic is glomerular filtration, renal blood flow and their control. Is there any point to discuss? This is renal plasma flow, 625 ml per minute coming to the afferent arteriole, getting filtered. Now this value, glomerular filtration rate, is 125 ml per minute. So how will you define? Most common question asked in your viva. जो अपना ग्रैंड वाइवा होता है ना, एक तो होता है आपका प्रैक्टिकल वाइवा, टेबल वाइवा, एक होता है आपका ग्रैंड वाइवा. So the most common question asked is define GFR, give its normal value. It is the amount of filtrate which is getting formed from both the kidneys or all the nephrons present in both the kidneys per minute or per day. Okay, amount of filtrate coming from blood and from both the kidneys, all the nephrons are involved, and per minute how much is getting filtered. That is GFR, glomerular filtration rate. So finally you can see 1 ml per minute. Urine formation rate is 1 ml per minute. ठीक है, ये गाइटन की वैल्यूज हैं. जब भी आपके एमसीक्यूज आएंगे या एमसीक्यूज के अंदर आपकी वैल्यूज आती हैं, तो यू कैन ओनली चैलेंज व्हेन यू आर आंसरिंग फ्रॉम गाइटन. Okay, another book for these MCQ or values are Ganong, but Ganong is a review book. For review, you have to have a view. So that is for pre-PG purpose. Ganong is a review book, review textbook of physiology. But for your level, it is Guyton. So in Guyton, सबसे पीछे पेज पे सारी वैल्यूज दे रखी हैं. आप किसी भी बुक से पढ़ो, ठीक है, पर आप उन वैल्यूज की या तो फोटो ले लो या प्रिंट निकालो और उसी वैल्यूज को आप इम्प्लाई करो. ठीक है, आप तभी चैलेंज कर सकते हो जब आपकी वैल्यूज गाइडेंस से है. द मोस्ट ऑथेंटिक बुक फॉर दीज वैल्यूज, किसी भी चीज की वैल्यू फिजियोलॉजी में बात करें तो गाइडेंस की मानिए. What is filtration fraction? It is GFR upon renal plasma flow. That gives you the value of 0.2. ये points आपको पता होने चाहिए. What do you mean by GFR? What is filtration fraction? Glomerular filtrate composition. What is it? Plasma excess. What is filtered is except formed elements and plasma protein, all is getting filtered. Except plasma proteins and formed elements, all are getting filtered. Okay, what are the formed elements? RBC, WBC and platelets. So when urine analysis is done, if RBCs appear, it is not a good sign. Until unless in case of females, if the urine analysis is done during the menstrual period, the RBCs does not appear in the urine analysis. Okay, so glomerular filtrate composition is about the same as plasma except the formed element in the plasma protein. Filtration fraction is GFR upon renal plasma flow. It is 625 ml per minute, and that is 20% of the plasma is getting filtered. Now the combination of this, इसने कहा था ना कि filtration 2 बार हो रहा है, किसने कहा था? तो फिल्ट्रेशन एक ही बार होगा. अब इसको समझने के लिए फिल्ट्रेटिंग मेम्ब्रेन का स्ट्रक्चर पता होना चाहिए, और हम क्यों कह रहे हैं कि प्लाज्मा प्रोटीन फिल्टर नहीं होगा. आदित्य क्यों नहीं होगा? क्यों नहीं होगा प्लाज्मा प्रोटीन फिल्टर? साइज और साइज हो गया एक, ठीक है. दूसरा क्या होता है कि जो फिल्ट्रेटिंग मेम्ब्रेन का स्ट्रक्चर है उसके अंदर कोलेजन फाइबर्स और नेफ्रिन, ठीक है, प्रोटीन स्ट्रक्चर्स प्रेजेंट होते हैं. तो दोनों का के अंदर क्या हो जाता है, नेगेटिव चार्ज होते हैं. तो नेगेटिव चार्ज क्या करेंगे, एक दूसरे को अपोज करेंगे. तो अलोंग विद द साइज ऑफ द प्लाज्मा प्रोटीन, another important factor which is responsible for repulsion or avoidance of filtration of plasma protein in the filtrate is the negatively charge present in the plasma protein as well as the negative charge present in the filtrating membrane. What do you mean by the filtrating membrane? It is the endothelium lining of the glomerular capillaries, okay, basement membrane plus the visceral layer of the Bowman's capsule. Here, what is Bowman's capsule? It is the blind beginning of the nephron. It is a funnel-shaped structure, and this blind structure, okay, it has two layers, the visceral layer and the parietal layer. The visceral layer, the basement membrane, and the endothelium lining of the capillaries results into the formation of the filtrating membrane. The space between the visceral and the parietal layer results into the Bowman's space. So the renal plasma flow which is coming into the Bowman's capsule is getting filtered and entering as filtrate into the Bowman's space, finally into the proximal convoluted tubule. So what is the structure of the filtrating membrane? It is the endothelium lining of the capillaries. Now what is the special property of this endothelium? It has slits or pores. तो इसके जो slits and pores होते हैं 50 to 100 nanometer dimensions के होते हैं. Then comes the basement membrane, okay, उसके और भी कम होंगे. Then comes the epithelium lining of the Bowman's capsule, that is the visceral layer. It is getting formed of the podocytes, and the foot process of the podocytes, they are known as pedicels, and these pedicels, they are slits or small spaces that will only allow 5 to 10 nanometer dimensions उसके ही molecules को enter होने देंगे. और जो basement membrane है और जो capillary जो epithelium lining है Bowman's space की, उसके अंदर collagen भी है, nephrin भी है, और भी proteins present हैं. तो वो सारे प्रोटीन्स नेगेटिवली चार्ज हैं, तो वो प्लाज्मा प्रोटीन्स को अलाउ नहीं करेंगे एंटर होने के लिए, और कोई भी फॉर्म्ड एलिमेंट एंटर नहीं होगा. So if there is problem in the filtrating membrane, definitely it is going to affect the glomerular filtration rate. So in some cases congenital defect is there in which there is change in the structure of the filtrating membrane that results into the appearance of proteins in the urine, proteinuria it is known as, and that results, in that case the protein appears in the urine. ठीक है, filtrating membrane एक बहुत constant value देता है अपने को, that is known as filtration coefficient, that will help us to calculate or to deduct the formula of glomerular filtration rate. तो केवल ये एक 3 कंपोनेंट से बनी हुई फिल्ट्रेटिंग मेम्ब्रेन जो है वो फिल्ट्रेशन कोएफिशिएंट वैल्यू देती है। फिल्ट्रेशन कोएफिशिएंट की वैल्यू किससे मिलती है? टोटल सरफेस एरिया ऑफ द फिल्ट्रेटिंग मेम्ब्रेन एंड thickness of the filtrating membrane, that is calculated, and the constant value that comes out of this filtration coefficient or Kf is 12.5. That is 12.5. So if the questions come, what are the factors which will be responsible for change in GFR? First is filtrating membrane. ठीक है? तो जो स्ट्रक्चर इसके लिए रिस्पॉन्सिबल है फिल्ट्रेशन के लिए, उसके अंदर अगर कोई प्रॉब्लम होगी तो डेफिनेटली देयर विल बी चेंज इन द ग्लोमेरुलर फिल्ट्रेशन रेट। इसका भी सिर दिख रहा है क्या? आ जाओ, आगे आ जाओ, कम इन फ्रंट। तुमसे पीछे वाला वो बेचारा गिल्ट में खड़ा हो गया। ब्लू शर्ट इसके पास, तुम्हारे पास है। आ जाओ, आगे आ जाओ बच्चे। वेरी डिफिकल्ट डेज इन हॉस्टल, कि रात में सोने नहीं देते, है ना? योर बायोलॉजिकल क्लॉक इज वेरी मच डिस्टर्ब, स्लीप पैटर्न इज हाईली डिस्टर्ब, नॉर्मल कुछ नहीं रहा, सब आर्टिफिशियल इंटेलिजेंस हो चुकी है, नेचुरल कुछ नहीं रही। Effect of size and electrical charge of dextron on filtrability of the glomerular capillaries. Okay, ये आपके शायद नहीं आए इतना डिटेल में। So what are the methods to estimate glomerular filtration rate? ठीक है, तो glomerular filtration rate जो होता है वो basically, okay, it depends on two important things. One is Kf, that is filtration coefficient multiplied by net effective pressure. Okay, what do you mean by Starling forces? You might have studied in plasma protein section what are Starling forces. What is the most important function performed by plasma protein? Maintenance of colloidal osmotic pressure. This maintenance of colloidal osmotic pressure is done with the help of filtration and reabsorption. One end of the capillaries, they are responsible for filtration, and the other end is responsible for reabsorption. So similarly, the pressure they are acting in this scenario, that is resulting into net effective pressure. Clear? What do you mean by hydrostatic pressure, and what do you mean by arterial blood pressure or blood pressure? Is there any difference between hydrostatic pressure and blood pressure? This is the method of estimation of GFR, okay, glomerular filtration rate. So what are the conditions that we have to see if we are going for method of estimation of GFR? So these points has to be kept in mind that they are freely filterable at the glomerulus. उनको filtration में कोई problem नहीं है. Neither they are getting deabsorbed, neither they are getting secreted, not synthesized by the tubule, not broken down by the tubule. So the most common example comes out to be creatinine. Okay, creatinine is in vivo, and if you have to infuse and do, then it is para-aminobutyric acid. Okay, now किस पे बात कर रहे थे अपन? GFR के net effective filtration pressure. Okay, कैपिलरीज का ब्लड प्रेशर कितना होता है? Is there any change in the pressure when we are talking from arteries, arterioles, venules? इनमें change होता है? होता है, ठीक है ना? In case of these glomerular capillaries, the hydrostatic pressure, what is hydrostatic pressure? It is the pressure exerted by any fluid But when we are talking of the glomerular capillaries, the fluid is blood. Okay. But when we are talking of hydrostatic pressure of the Bowman space, it is not blood, but it is fluid or the filtrate which is creating this pressure. So we are using the word hydrostatic pressure. So hydrostatic pressure is the pressure exerted by any fluid. But when we are talking of the glomerular capillaries, it is the blood which is present and which is creating the pressure, whereas in the filtrate formation or the filtrate which is present in the Bowman space, it is not blood, but it is only filtrate. So hydrostatic pressure of the filtrate present in the Bowman space. तो हमें 4 प्रेशर्स के बारे में बात करना पड़ता है. 4 में से 2 प्रेशर जो होते हैं वो फिल्ट्रेशन को फेवर करते हैं और 2 प्रेशर जो होते हैं वो फिल्ट्रेशन को रेजिस्ट करते हैं. जब अपन ग्लोमेरुलर कैपिलरीज की बात करें तो उसके अंदर क्या है, प्लाज्मा प्रोटीन. प्लाज्मा प्रोटीन का क्या फंक्शन था, मेंटेनेंस ऑफ कोलॉइडल ऑस्मोटिक प्रेशर और मेंटेनेंस ऑफ ऑस्मोलरिटी. सो प्लाज्मा प्रोटीन रेजिस्ट filtration, so they are opposing the filtration, whereas the pressure, blood pressure or the hydrostatic pressure of the capillaries, they are 60 millimeters of mercury and they will favor filtration. So hydrostatic pressure of the glomerular capillaries, they are responsible for filtration, whereas the protein pressure or the plasma protein pressure, okay, that is resisting or opposing the filtration. तो 2 प्रेशर जो ग्लोमेरुलर कैपिलरीज के अंदर एक्ट कर रहे हैं वो कौन कौन से हो गए, हाइड्रोस्टैटिक प्रेशर और कोलॉइडल ऑस्मोटिक प्रेशर. कोलॉइडल ऑस्मोटिक प्रेशर किसकी वजह से होता है, मेनली एल्ब्यूमिन की वजह से होता है. और एल्ब्यूमिन का जो प्रेशर है वो रेजिस्ट करेगा फिल्ट्रेशन को, पर कैपिलरी हाइड्रोस्टैटिक प्रेशर और कैपिलरी ब्लड प्रेशर, ओके, of the Bowman glomerular capillary is 60 millimeters of mercury, which is not present elsewhere in the body, is responsible for filtration. Now whatever the blood or the plasma is getting filtered, it is entering into the Bowman space. Now this Bowman space has also pressure, पर इसके अंदर नॉर्मल हेल्दी कंडीशन में कोई भी प्रोटीन फिल्टर होके नहीं आएगा. So the colloidal osmotic pressure generated in the filtrate is 0. Okay, only what remains is the hydrostatic pressure in the Bowman space that is going to oppose filtration. So out of 4 pressures, only one pressure is responsible for filtration, that is the hydrostatic pressure of the glomerular capillary. इन सब का सम अप करके निकालते हैं तो फाइनल और नेट इफेक्टिव प्रेशर comes out to be 10 millimeters of mercury. That 10 millimeters of mercury is multiplied by filtration coefficient, that comes out to be 12.5. और filtration coefficient किससे कैलकुलेट किया हमने, टोटल सरफेस एरिया ऑफ द ग्लोमेरुलर कैपिलरीज एंड the width or the thickness of the filtrating membrane. उसकी एक कांस्टेंट वैल्यू आती है और वो कांस्टेंट वैल्यू होती है 12.5 और नेट इफेक्टिव प्रेशर कितना हो जाता है 10. तो हम कैसे कैलकुलेट करें, 10 into 12.5 फाइनली रिजल्टिंग इनटू 125 ml per minute of GFR. Okay, so glomerular filtration rate per minute is 125 ml per minute. When we are talking per day, it is 180 liter per day. कितना टाइम हो गया? अच्छा, तो नेक्स्ट में अपन देखेंगे कि फैक्टर्स जो कि GFR पे इफेक्ट करेंगे. ठीक है, what are the factors which are going to affect the glomerular filtration rate? ठीक है, so the story will start from the diameter of the efferent and the blood pressure value. चलो जक्टा ग्लोमेरुलर एपरेटस क्या होता है ये खत्म करते हैं अपन. क्या होता है, मेरे को लग रहा है तुम पढ़ने लग गए इससे पूछ रही हूँ. चलो शॉर्ट नोट आता है juxtaglomerular apparatus पे, ठीक है. So the juxtaglomerular apparatus is why we are calling it apparatus, because it is getting formed from the three main components. And what are the three main components? They are the juxtaglomerular cells, okay, macula densa and lacis cells. Okay, what are the glomerular juxtaglomerular cells? उसको अपन juxta बोल रहे हैं क्योंकि वो near the glomerulus है. कहाँ पे present होते हैं? tunica media of the afferent arterioles. Histological picture of that particular area changes. It becomes granular in appearance because it is mainly carrying the renin inside it. The granules are present of renin. It gives granular appearance. The shape changes, okay, make a cushion-like appearance. So they are the first component of juxtaglomerular apparatus. Second component is macula densa. Now again the histology picture changes of this particular section of the distal convoluted tubule, क्योंकि इनका function जो होता है, they are acting as chemoreceptors. They are acting as chemoreceptors. Why we are calling it them as chemoreceptors? Because they are sensitizing the presence of sodium and chloride ions. Clear? Extraglomerular जो होता है, बीच के लेसिस सेल्स जो होते हैं, वो mesangial cells होते हैं. They are responsible for the contractile nature of the glomerular capillaries, and these are responsible for the secretion of renin, and renin finally helps into the control of the blood pressure. So what are the factors which will trigger the release of renin? Either there is decrease in the blood volume, okay, either there is decrease in blood pressure, either there is change in the sodium or chloride ion concentration, okay, or the stimulation of sympathetic nervous system. So 4 factors responsible हैं जो कि stimulate करके renin को release करेंगे. फिर renin का अपना function है, that is a separate story, we'll do it later on, but just what is the structure of juxtaglomerular apparatus, what all things are getting involved, and what is the role. The afferent arteriole is acting as baroreceptors. They will detect the change in the blood pressure. So they are known as baroreceptors. Macula densa, they are acting as chemoreceptors because they will sensitize the change in the chloride and sodium concentration. Okay. So next we'll take factors affecting GFR.